Okay, let's consider the remaining factors that govern microevolution. Okay, we've talked extensively about mutations in previous classes. We talked about natural selection in the last video, and now we're going to talk about genetic drift, migration, and non-random mating. So this idea of genetic drift is that there's a change in the frequencies of alleles in a population due to random fluctuations. There are two ways that this is normally occur in nature that we will discuss in subsequent slides. But when genetic drift happens, it can lead to a loss of alleles. So here you see we had genetic drift and we're left with only red, or here we're left with only blue. It can also lead to a fixation of it. So here we have almost equal numbers of both. These alleles that it's acting on or causing to increase or decrease can be deleterious, neutral, or beneficial, right? So here it doesn't matter so much because it's random. So by chance, it looked like this, or by chance, it looked like that, okay? And how it ends up, that rate of change depends on the population size and the initial frequencies. So like if you have all red, this is your initial allele frequency. Over time, you expect to keep more of the same unless something happens, right? And so one type of genetic drift is this bottleneck. So think if you had a different color marbles in a bottle and you shook them, what would come out, okay? Would it be similar to the original population, or would it be something totally different? So this is a genetic drift that occurs when most members of a population are limited randomly. Okay, it doesn't matter, oh, all of this genotype died out. No, it's saying some random event caused certain members to just die. This can usually be natural catastrophe, so an earthquake hit, a flood, a drought, or it can be human destruction where someone went in and got rid of most of the land where the animals lived. It could be fire, a thing that wiped out their habitat. Okay, so something happened and it wiped out members of the population. So what you get, as you can see here, is less variation. So up here there are more variation, more different color balls than you have in the remaining population. So now when this population goes on to reproduce, they can only reproduce with individuals who look like them. So essentially we lose red. Okay, we lose this very light color here. Those are gone from our population unless something happens where they migrate back in. Now, this happened to the African cheetah several years ago, and so that now the species that remain have very low genetic variation. Okay, so unless there's some way to introduce new organisms within a species that are going to increase that genetic diversity, that surviving population has less genetic variation. Another example of genetic drift is the founder effect. So with this one, there's still that change in our little frequency, but it's due to a geographical change. So maybe we had some ladybugs on the mainland. By chance, some of them got sent to the island, but it doesn't represent the original. But they are now separated from each other. And so this founding population here has less genetic variation than the original one. And the frequency, what is here, differs by chance. This could easily have been a yellow ladybug, right? But in this case, it was a red one. So this is by chance. The red one went on to found essentially a new population. Okay. Then there's migration, also gene flow. And this is really people or organisms are moving from one population to another. And when they do that, so if this was a closed population of all homozygous recessive individuals, and this was an isolated population of all homozygous dominant, and they just decided, I want to be over there. Or I got lost from my colony and I ended up over here, right? So the introduction of this new organism within this population brings with it new genetic variation. And so by doing so, once this Homo dom starts mating with this Homo recessive, we are now introducing the dominant allele, right? We are shifting the gene pool here because now there's going to be the possibility of the darker colored bugs. And so this is the idea of migration, where we have two well-established populations, but individuals going between the two are going to alter the gene pool. What happens when you do that? Well, it's going to reduce those differences. Here we were 100% dom and 100% recessive. We're going to reduce that. We're no longer going to be 100% dom here. And it's going to enhance or increase that genetic diversity, because now we're getting new genes within that population. And who knows what that could mean for reproductive success. Another idea to consider is non-random mating. Okay? So non-random mating is it's not a chance that you pick a mate. Okay? And so this is also called assortative mating. Okay? Assortative mating, which means you have a preference. Okay? So organisms or individuals are going to mate with who they are attracted to. So there's something called selection bias, where we know that there are organisms who are the prettiest, okay, the fastest, the strongest, tend to attract the mates. And so it can be positive or negative assortative. So if it's positive, organisms that are similar to each other. So organisms with the similar phenotypes tend to mate. Whereas if it's negative, individuals with different phenotypes tend to mate. Okay. This is again going to introduce more variability. This is individuals that are similar. Then you have inbreeding versus outbreeding. And I am almost certain that you know what that means. So inbreeding is like keeping it in the family. We have mating within familial units. So if we look at this pedigree, we can see like, okay, we have two cousins here who have reproduced together. And then outbreeding is where you mate with someone you're not related to. Okay. But it is important to note here that inbreeding and outbreeding don't affect the allele frequencies, but they do disrupt the balance of genotypes, okay? And we also know that the reason inbreeding is frowned upon is because it increases the likelihood of two individuals having the same recessive alleles, okay? This is great if you were breeding something like cattle or dogs and you wanted to make some prize animal. This is not so great when it comes to people, because every person carries with them recessive alleles, and when people in the family, the likelihood is that they carry these same recessive alleles, which may lead to a disorder or disease in their offspring, okay? And now, aside from that, so if you think back to our chart, we know that there was new genetic variation that's usually random by chance with the introduction of mutations. And then there's that existing genetic variation that we can act on. Okay, but moving forward, how do we then get new genetic variation, right? What are some sources of new genetic variation to introduce in today's society? Where is it going to come from? And I think the answer is where it's been coming from, right? So I'll also note here that when we talk about those mutations, they can be deleterious, they can be neutral, they can be beneficial. However, the likelihood is that they will usually be deleterious or neutral. Beneficial mutations are less common than the other two. I will also note here that neutral mutations are not acted upon by natural selection, because even though you're changing the sequence, you're not changing the protein, so you're not changing the trait or the gene, or not the gene, the trait or the protein or the overall individual. Okay, so there is no outward change. So that's not usually acted on by evolution or natural selection. But then how can we introduce these new variabilities? Okay, so these are all things we've talked about: independent assortment and Mendelian inheritance, crossing over during meiosis, prokaryotic gene transfer. We talked about 3 different ones that you should remember. Then things like gene duplications, chromosome number and structure mutations. These are all ways to introduce variability. Let's consider 2 in a little more detail. One is this horizontal gene transfer. This is, I guess, like the name occurs. So when we think about gene transfer, we're usually thinking in a vertical from parent to offspring. But now we're talking about horizontal. So not into your offspring, but to another individual that you can give genes to. So this can happen from prokaryotes to eukaryotes, eukaryotes to prokaryotes, prokaryote to prokaryote, eukaryote to eukaryote, okay? So it can happen within and among prokaryotes and eukaryotes. We talked extensively about how this happens in bacteria and how the end result is that bacteria having some new genetic variability, right? It can also then happen in other species. And how it happens depends on what two species we're talking about. But horizontal gene transfer is one way to introduce new genetic variation. Another way is this idea of repetitive sequences. So we know that in eukaryotes, we have tons of repetitive sequence, and we've talked about them. We classified them as unique or not repetitive, moderately repetitive, and highly repetitive. Well, let's then consider one thing we talked about before, transposons. And we know that transposons can move around the genome. Well, by doing so, if we're talking retrotransposition, we're making more copies. So that copy and paste mechanism. So each time we go through replication of a transposon, we're making more that then has to be inserted somewhere in our genome. Well, if my transposons are reproducing less than yours, we now have genetic variability because you have more copies of transposons than I have. Another thing that this gives us is where those transposons insert can be disrupting genes, which can then have a whole nother consequence. Now, similarly to transposons, we also have another repetitive sequence, or two more, called micro and mini satellites. They are distinguished by how long they are each individual and then as a unit. So if it's micro, it's 1 to 6 base pairs. And then they repeat in tandem repeats up until a few hundreds of base pairs long. And then if it's mini, it tends to be 60 to 80 base pairs, and then they repeat 1 to 20 kilobase pairs. An example of mini satellites are these telomeres. But remember, that's a range from 1 to 200 kilobase pairs. And you could have 1, I can have, 20. You can have 1, I can have 20, someone else can have 5. Those lengths make you different from me. Those introduce genetic variability. And so the importance of this is we can use this in DNA fingerprinting. Those micro and mini satellites can help us with gene mapping and then identifying population variation. And so we know that DNA fingerprinting, and we did the lab on this, that every person is different, right? As unique as an actual fingerprint. And it's dictated by those repeats that we have. So you can use this to identify suspects and to also identify ancestral and relations between two people. Okay, so are there any questions, comments, or concerns on this topic?